In this module, we will again talk about the inducers that induce a specific product during fermentation process. As we discussed in our previous module, that most of the inducers that need for the production of different enzymes, which we called as inducive enzymes. But in special cases, when fermentation process is deal with some recombinant products, or those proteins which we called as heterologous proteins, the meaning of this, in which we just incorporate the foreign gene into the microorganism, such as insulin gene into the bacterium or into the Pichia pastorius or a yeast, or any other expressed proteins from a foreign source, foreign gene source, such as if we want to produce any desired protein that is not basically the part of that natural system, we incorporate, we have a gene cloning and gen cloned organism, and we want to produce, and we have to express that protein, which is known as the heterologous protein, then we need a specific inducers. Sometimes those inducers are also known as promoters. So when we design the whole system that has a specific inducers or a promoters. Actually, when we say that, when we talk about those heterologous proteins, that either they are not produced when the normal system of that cell is going on, or in other hand, that those product is inhibited by different factors of that organism. So that's why we have to incorporate a specific inducers or the promoters just to have the expression of those specific proteins. So there are so many examples of that. So that only we can achieve the production of those products only in those proteins which we called as heterologous protein, only in the presence of those promoters. So as concerned, the optimum growth conditions that may not be synthesized those heterologous proteins, so only inducing after that we can have the chance to produce those, as I have already told you. So here you can see in one example, so using Saccharomyces cerevisiae, where GAL1, mean galactoside is 1, promoter is a part of the gene expression system. Product formation may be induced by the presence of galactose. So addition to the growth medium that basically control that contain the glycerol or the low non-repressing level of the glucose as a carbon source. In this case, you can see here that addition of the glycerol that act as the inducer. So in other case, in case of two other yeast, there is an alternative system, and then the system because of the presence of alcohol oxidase promoters. So that there are different promoters. So that is only we have to select what kind of the promoter we have inserted while making the clone. So while expressing that protein, we should add that promoter on a specific time in the fermentation medium. So during the growth, on which sometimes the methanol are used as an inducer, sometimes IPTG is used for different protein expression. So there are number of promoters. So when we are dealing with the cloning and in a genetic engineering experiment, while cloning of different genes into the microorganism having the fast growth, then we have to select the specific promoters or the inducers, and then we have to induce that product or that especially the proteins. So we have to add on a specific, in case of the IPT, so we only add into the medium the IPTG when there is a specific level of growth. So then when we, in the start, we have a threshold of that organism, then we add the promoter. So after the addition of that promoter, then the expression or the production of that heterologous protein start to produced.